Okay, let's see what notes you've made around this information about cyclooxidase enzyme. So we read cyclooxidase is one of the enzymes needed in the formation of prostaglandins. So making some assumptions here, cyclooxidase, if it's an enzyme, we know therefore it must be a globular protein. If it's a protein, we know that the monomers are amino acids, that it has a specific 3D shape, and that will be due to its tertiary structure. We know that it's going to be affected by changes in temperature, pH, concentration of substrate, and things like that. So lots of assumptions here that we've made about cyclooxidase, even though we've never heard of this specific enzyme before. We read that it was needed in the formation of prostaglandins, so I've done some research here and found out that prostaglandins are actually lipid compounds and they're derived from fatty acids, that they have hormone-like effects within the body and they're found in almost every tissue in animals. So this is something that's not actually on the specification, so we can assume that the actual structure of prostaglandins and their effects aren't that important for the question. It already tells you that prostaglandins are compounds that are involved in causing fever, pain and inflammation. And just because we don't know at this point what's relevant and what isn't, I've made a note about what each of these things actually mean. So fever is when your body temperature rises above the norm. And pain is an unpleasant feeling caused by intense stimuli. And if you're actually an A2 student who's already studied nerves in F214, well then you will know that pain is also a result of action potentials, in particular an increased frequency of action potentials. If you remember, that's how your brain perceives strength of stimuli. And inflammation is actually a protective response within your tissues, and it's there to initiate repair due to specific damage. So the question goes on to explain that the cyclooxidase enzyme catalyses the conversion of arachidonate to prostaglandin H2. So over here, just because it's really, really important that we get this information straight in our heads, I have set out this specific reaction. So the substrate here that cyclooxidase enzyme is catalysing this substrate, converting it into the product prostaglandin H2. And we notice that this is actually the second reaction within a pathway that actually links three reactions. And the first was the catalysation of phospholipids into arachidonate by an enzyme that they've not actually named within this figure, but we've called it enzyme A. So again, just to make it very clear in our heads, the phospholipid is the substrate of this enzyme and the product is arachidonate, which actually becomes the substrate for the cyclooxidase enzyme. So this product actually determines whether if this enzyme has a substrate to work or not. And then the prostaglandin H2 is converted by a third enzyme now into prostaglandin. And again, that enzyme wasn't named, but we know that the substrate for this enzyme is prostaglandin H2, and the final product there is prostaglandin. So this is the full pathway here. The phospholipid being converted into arachidonate, which is by the first enzyme, and this arachidonate becomes the substrate for the COX enzyme, which is then converted into prostaglandin H2. So this is the second reaction, but the first has to happen before so that the COX enzyme actually has a substrate to convert. Likewise, if this prostaglandin H2 isn't created, well then the third enzyme doesn't have its substrate. So prostaglandin H2 converted into the final product, prostaglandin there. And further down then, it says the COX enzyme is found attached to the inner surface of the endoplasmic reticulum. Now, endoplasmic reticulum is something that we saw in F211 when we did eukaryotic structure. The two forms, rough and smooth, So if we remember, rough endoplasmic reticulum is called rough because it looks rough due to it having ribosomes attached, and its function is protein synthesis and transport. And smooth endoplasmic reticulum looks smooth because there are no enzymes attached, and the function of this type is lipid synthesis and transport. So looking at this diagram, there's no obvious ribosomes attached here, so we could assume that this is smooth endoplasmic reticulum involved in lipid synthesis and transport rather than rough endoplasmic reticulum. And this is just a bit of extra information because I don't know if it's going to be useful or not. Just a reminder that endoplasmic reticulum is found in eukaryotic cells, and these are cells with a true nucleus that contain membrane-bound organelles, such as the endoplasmic reticulum. So other things that have been noted on this diagram is the fact that all of this, on either side of the endoplasmic reticulum, on the outside of it, is cytoplasm, and this is where other organelles will be present. So organelles such as the Golgi apparatus, you've got your lysosomes, more endoplasmic reticulum, mitochondria, chloroplasts if we're talking about a plant cell, various other organelles. Okay, so this is the position of the COX enzyme attached to the inner membrane. And just a reminder that membranes are made of phospholipids arranged in a bilayer. And this is what one phospholipid looks like. So this is the head of the phospholipid, which is actually made of a phosphate group and glycerol. We call it a hydrophilic head because it's water-liking. And then we have two tails, and we call this bit the hydrophobic tail. They're actually two fatty acid molecules, hydrophobic because they tend to repel water. Now, as I'm thinking about this, I remember now phospholipids were mentioned up here. Phospholipids were the substrate for this first reaction, and if we don't actually have phospholipids present, well then we can't create this product, arachidonate, which is actually the substrate for the COX enzyme. So this potentially could be important for this question. And just as a bit of extra revision then, so I've drawn this diagram just as a reminder as to how these phospholipids are arranged. So all of the tails being hydrophilic, they face away from water, and all of the tails being hydrophobic, they face away from the water. So they all align themselves inwards. And it is an alignment. They're not physically bound to each other. They just align next to each other due to the properties. So the heads there being hydrophilic and the tails being hydrophobic. They don't physically form bonds. So this is all the information that I took from this information before attempting the question.